everyone, welcome to sbiology. In the previous class we have discussed about the Michelson and Stall's experiment for semi-conservative DNA replication. Today let's discuss about the replication process of DNA or DNA replication process. DNA replication takes place in the S phase of cell cycle. Okay. Cell cycle ro S phase re DNA replication process ହେଉଛି. So what is DNA replication? We have already discussed. DNA replication is a process in which two identical copies of daughter double stranded DNA form from a parental double stranded DNA. Okay. DNA replication होची गोटे प्रोसेस जहा द्वारा गोटाए डबल स्टैंडर्ड डीएनए रु दुईटा आइडेंटिकल डॉटर डबल स्टैंडर्ड डीएनए फॉर्म होची. Michelson and Stahl also proposed an experimental proof for DNA replication, that is semi-conservative DNA replication, in which the double stranded DNA would separate first. Suppose this is a double stranded DNA. In the process of replication, two strands are separated out first. Okay. And both the strands are act as template. Okay. And newly synthesized strands are paired with them to form two new daughter DNA. Daughter DNA. In this process, the parental strands are conserved half. Okay. The daughter DNAs are contain one strand from parent, one strand from newly synthesized strand. Okay. So this is the DNA replication, that is semi-conservative DNA replication. DNA replication includes some enzymes, or DNA replication requires some enzymes to complete the process. Okay. So different enzymes involved in DNA replication process. What are these enzymes? Let's see. First, DNA helicase, DNA polymerase, trimase, single strand binding proteins, in short form SSBP, topoisomerase and DNA ligase. DNA replication process अनेक गुड़े एंजाइम दरकार करुछि. काहेकि एंजाइम्स आर बायोलॉजिकल कैटलिस्ट. एंजाइम्स एक्सेलरेट द बायोलॉजिकल रिएक्शन. जे कोनोसी बायोलॉजिकल रिएक्शन होछि ताको एक्सेलरेट करुछि किए एंजाइम. तो सेति पई कि जे कोनोसी बायोलॉजिकल रिएक्शन बॉडी भीतर होछि किछि एंजाइम दरकार होछि. रेप्लिकेशन इज आल्सो ए बायोलॉजिकल रिएक्शन and DNA replication involve different type of enzymes. DNA replication केते गुड़िए एंजाइम दरकार करूछि. प्रोसेस टाकू कंप्लीट करिया पई कि DNA replication process कंप्लीट करिया पई कि कोटिए DNA रु दुईटा DNA सृष्टि करिबा पई कि DNA कोन करूछि केतेटा एंजाइम दरकार करूछि. ए डबल स्टैंडर्ड DNA contains two strands of DNA. Okay. And each strand contains one 5' end and one 3' end. Each strand have one 5' end and one 3' end. And we have already discussed that two strands are opposite in direction. It means anti-parallel. When one strand starts from 5' and end at 3' end, another strand starts with 3' end and end with the 5' end. Okay. So two strands are anti-parallel in nature, and each strands are attached by hydrogen bonds. Okay. And each strand contains nitrogen bases, that is guanine, cytosine, adenine and thymine. Okay. Guanine always attached to the cytosine of another strand, and cytosine of one strand always attached to the guanine of another strand, and thymine of one strand always attached to the adenine of another strand. And adenine, again vice versa, adenine can add to thymine of another strand, and guanine added to cytosine through triple bond, through 3 hydrogen bond, and adenine can attach to the thymine through double bond. Okay. When there is a adenine, there is also a thymine. When there is guanine, there is also a cytosine. Okay. And the guanines are attached to the cytosine through 3 hydrogen bond, and adenines are attached to the thymine through 2 hydrogen bond. Okay. Let's see the steps of DNA replication. First, identification of the origin of replication. Okay. Identification of the origin of replication. प्रथम step है कौन ना identification of origin of replication. Origin means the place where the replication would start. Okay. There is a point from where the replication would start. That is known as origin of replication, or in the short form it is known as ORI. Okay. In case of prokaryotic, origin of replication is 1. In case of prokaryotic cells, or in case of E. coli, origin of replication is single or 1. That is known as ORIC. Okay. But in case of eukaryotics, origin of replications are more than 1. Okay. Next step, unwinding of double standard DNA to single standard DNA template. I have already told that when the replication process starts fast, the double standard DNA would separate, and both the strands act as template for newly synthesized strand. Okay. The next step is unwinding of double standard DNA to single standard DNA template. Next step, formation of replication fork. What is replication fork? We will discuss it later. Next step, initiation of DNA synthesis and elongation. And the last step is removal of primers and reconstitutions of chromatin fiber. Which point of a double standard DNA is known as origin of replication? The point which is rich in adenine and thymine is known as the origin of replication. Okay. गोटे पॉइंट डबल स्टैंडर्ड डीएनए रो गोटे पॉइंट जोठी अधिका मात्रा रे एडेनाइन आउ थायमिन अछंती सेही पॉइंट टाको कहै जाउची कोन ओरिजिन ऑफ रेप्लिकेशन. Let's see briefly the DNA replication process. Each strands of DNA made up of 4 nitrogen bases, that is adenine, thymine, cytosine and guanine. 2 strands are complementary to each other. It means wherever T in one strand, there will be A on another strand, and wherever C in one strand, there will be a G in another strand. Each strand have 1 5' and 3' end. 2 strands are run on opposite direction. So, in the first step of DNA replication, separation of double stranded DNA occurs. First, the double stranded DNA would separate and form a Y-shaped structure that is known as the replication fork. This replication fork is made by an enzyme called helicase. Helicase unzips or separate the double stranded DNA into 2 template strands. After that, separated strands act as template for synthesis of new DNA strands. In the second step of DNA replication, an enzyme called primase come and add at the specific point, that is origin of replication, and starts to form primer. Primer means small segment of RNA. This makes the starting point of replication. In the third step,
Another enzyme called DNA polymerase 3 comes and bind to the primer and forms new bases of DNA. DNA polymerase 3 can move on the direction 5' to 3' only. So, one of the template strand which runs from 3' to 5' direction is called as the leading strand, where the DNA polymerase 3 continuously make the new strand. And another template strand of DNA which runs on the direction 5' to 3' is called as the lagging strand because it runs in opposite direction. So, DNA polymerase 3 cannot make strands continuously because it is in opposite direction. So, here DNA polymerase 3 makes strands in small fragments, which is known as the Okazaki fragments. Each fragment is started with RNA primer or primase from 5' to 3' direction or 5' to 3' direction. In the fourth step, once the DNA strands are made, another enzyme called DNA polymerase 1, which is an exonuclease, come and remove the RNA segments or remove the primers by adding the DNA segments there. In last step, finally, another enzyme come which is known as the DNA ligase, seals the gaps or joins the fragments of the lagging strand, that is the Okazaki fragments, to form a continuous DNA strand. After that, two double stranded new DNAs are formed, and this process is called as semi-conservative DNA replication.
This is all about the DNA replication process. In the next video, in the next class, we will discuss about transcription of DNA. Till then, keep watching and keep learning. Don't forget to subscribe my channel, that is sbiology, for more video, and follow me on Facebook for more video. Thank you.